Hello everybody, my name is Bobby Cole, and this is the Cold Hard Truth. Here with me today I have Dr. Scotty with me, professional microbiologist.
Thank you.
How are you doing today?
I'm doing pretty good. How about yourself?
I mean, pretty good, thanks to the sponsor of today's episode, Scobie Snacks Kombucha. Get yours today. Thanks for sponsoring this episode. So, I've been hearing a lot about this CRISPR thing, right?
Yeah.
Can you maybe tell us a little bit about that? I haven't heard much about CRISPR, like what it is.
Yeah, so CRISPR was one of those things that was talked about a long time ago and had all these promises, and then people just, they kind of forgot as it just wasn't relevant. Basically, it's a gene editing protein. The way that it works, right? So CRISPR, and specifically with what we're working with today in our current research, is Cas9. Think of it as like micro scissors almost. We found it in bacteria, and what it does is, like, say a pathogen tries to infect this bacteria and stuff like that.
Like viruses and stuff like that?
Exactly.
Okay.
Think of it like a search dog, right? You give it a piece of DNA, kind of like you give a search dog scent, and then it goes after it. So bacteria gets infected with a pathogen, Cas9 locks onto it, so that next time it gets infected, it just chops it up into bits.
So CRISPR is a narc, that's what I'm hearing.
Basically, micro narc, yeah.
Yeah.
Our current breakthrough with it is with sickle cell anemia, and we've seen a crazy amount of progress with that. We have not just Cas9, but we also have another gene editing thing that works with it as well, which is Lyfgenia. And so both of these are used right now to essentially cure sickle cell anemia.
How does it do that? Like, if it's like a narc, is it like finding all the blood cells or something?
Yeah, so with Cas9, they basically take your blood, right? And this works outside of the body. They take your blood, you go do what you're gonna do, while they're basically giving that genome of sickle cell anemia to Cas9, giving the scent to the dog, and it clips up basically that specific genome. They give it back to you. Essentially, that's it for Cas9. Then you're cured of it.
I mean, if it's just the one blood sample, how does that help? Or does it like spread to other blood cells as well?
So there's a lot of blood transfusion and chemotherapy that is involved with this process of treatment. And Cas9 goes in, cuts out the sickle cell anemia genome, and through kind of a more like roundabout process, comes back, and essentially you have somewhat normal blood that is not infected by this genome.
So like slowly replacing all the blood.
Pretty much, yeah. And then for Lyfgenia, that is a lentiviral vector, which
That's a lot of words.
Yeah. To put it simply, think of a virus, right? And it inserts its DNA into your cells and whatnot. And with a lentivirus or lentiviral vector, think of like an empty virus, essentially.
Okay.
They plug any gene that you want in there, and it replaces it. Yeah, it almost silences the silencer. If sickle cell anemia was a silencer, right?
Okay. Yeah. So I guess why are people in an uproar about it? I mean, like, what are we looking forward to?
There's a lot to look forward to, because if you can just edit genes, and we can essentially edit any gene, so that would mean, like, for example, we can help with cancer, other genetic illnesses, and even just reconstructing what makes you, you.
I mean, I guess, hold on. Like cancer, I mean, what are we looking at there?
You have your immune cells, right? And they're trying to track down for cancer cells and whatnot. And unfortunately, right now, they have a problem with that, which is why people have cancer. If we could use CRISPR in order to change how those immune cells work to better target cancer and get rid of it.
So you give it the narc, you give it the dog.
The search
and,
dog,
yeah,
exactly.
and it can find those. Okay. What was that other thing you were talking about though?
I think it was just like other genetic diseases, and then also just changing your overall DNA makeup.
Oh, okay, so with that changing your DNA makeup, like,
Yeah.
What's the limits to that? I mean,
So from what we have right now in the scientific literature, in theory, it could be anything from changing the genomes for eyesight so that you can have perfect vision, up to making crazy different kind of modifications to your overall genetics to be able to survive in different climates even. The sky's the limit,
yeah.
in theory.
I mean, I guess this is kind of a bit of an off topic, but like you gotta stick to reality with it to a degree. Because I mean, the common complaint in films is like, oh, those wings on a human would never work. They'd have to be like 20 feet long or something.
It would be a gradual process. Think of it like how we see like 1970s computers or any sort of older technology.
Yeah.
We thought that touch screens and quantum computers were like totally futuristic, unrealistic. There's no way I could see my grandma from 900 miles away over a screen, but now that's reality.
Yeah, I mean, I've also heard people getting in an uproar over CRISPR though, with that being able to like edit people and ending up in the wrong hands and all that goodness.
There's definitely a lot of discussion about that when it comes to unborn human rights, or even things like agriculture, like livestock and plant life, because when you're talking about DNA, this does not just stop with humans. This can go all across the board. Once the door opens and you have this knowledge of things that you can prevent, why would you deny someone that? Why would you allow them in order to be born with these terrible diseases and whatnot?
I mean, Jamie, you've worked with like sheep and cows before, right? What do you know about like all this stuff? Yeah.
Well,
Everyone.
Scotty.
Special guest, Jamie, the editor here.
Yeah, I got to get out from behind the desk, get in the spotlight a little bit. But yeah, I've worked in the livestock industry my whole life before starting up here as a producer. And what I've seen in the livestock industry is some really interesting achievements done by this CRISPR-Cas9 technology. We've seen cattle get coated with a bacteriophage 1 protein that inserts a gene to withstand tuberculosis infections, and with that, that saves a rancher hundreds of thousands of dollars on some of these high quality breeding stock that he has.
Sure.
Now the other side of that is that those cattle cannot be sold as non-GMO or organic, which those two classes of product can create up to a 50% premium per pound, which is a really big deal to keep your rancher and all of our ag producers in business. With the growing population, we're projected to be close to 9.6 billion in 2050.
Yeah.
And we're losing a lot of farmland at the same time. So without these genetic modifications happening, we're going to see a huge collapse in our food systems. And so that demand of crops is going to go up. Since 2005, it's probably gone up about 50%. And then by 2050, it's going to be another 50%. Without having something like CRISPR to help make crops withstand harder conditions and yield more per acre, we're just not going to be able to maintain our society the way it is today. I mean, there's huge benefits when it comes to that. And I think it's ethically essential that as leaders of our country, we're all part of a democracy, that we need to support our agriculturists' ability to use these genetic modifying programs to feed us in an available manner. And not everyone's be able to pay for these high price non-GMO meats and other crop products with less ground, with less time.
This can also extend to the metabolism of animals and you and stuff like that, where you change the genes of your metabolic needs to be decreased so that you don't need as much to live. It kind of goes into the same vein as we're gonna have a higher population. We're gonna need more land and stuff like that. Now, if we have CRISPR that mitigates that as well as our own metabolism, then you would have less metabolic needs. So, yes.
Sorry to jump in on you.
Oh, it's okay. Go ahead.
So are you talking about like changing humans' metabolism, or are you talking about changing how animals grow? Because like I feel like there's kind of a difference between those two. You know what I mean?
It's all across the board. You can do all of it, at least in theory. And I mean, there's some research on it right now. Needs to be further studied, but you can increase the size of an animal and decrease their metabolic needs. More for less.
And you can also do this with plants as well.
Absolutely.
Yeah. And so we run into a lot less of an ethical issue when it comes to plants. We need to respect everyone's beliefs. So we need to produce organic, not GMO food for our nation for those who need it. And then we need to produce these metabolically changed animals and plants. Maybe there's an ethical issue there, but there's also an ethical issue of not feeding your people, right?
With respect to people's beliefs, like making the banana what it is today, we had to crossbreed and stuff like that to make the banana rather than being full of seeds and not a lot of produce.
Yeah, and this is completely true that domestication has completely changed a lot of our animal species as they would be in the wild. But even like our domesticated animals now, they can still breed and reproduce with their wild counterparts. A lot of them can. And they haven't changed to the point where they're unrecognizable either. Now, a pug might be a good example of unrecognizable compared to, you know, Canis lupus, but there was no way that our world was going to sustain us as hunter-gatherers since we've kind of came across that point. You get, you know, your sperm and egg cells outside of the body, you could change the offspring's genome. I don't know a whole lot about this CRISPR, but I don't think that anything can be perfect.
Are you asking just on the drawbacks of CRISPR?
Yeah.
Back to sickle cell anemia, right? You do have to go through transfusions, chemotherapy, and yes, right now it is expensive for those things. The more that is invested into CRISPR and any innovation really, the costs will cut down. They were looking at some things like, because I get it, people would be concerned about what if you cut the wrong things, like with Cas9, or what if you have a mutation. Well, currently the research states that if you don't give it the wrong thing, that it just won't do it. You have to deliberately give it a genome to cut. Right now we have sickle cell anemia taking place, like the treatment of it with CRISPR is taken outside of the body. So you don't have these loose gene editing tools running throughout your system. You also have the mutation aspect of it, or the cancer even. And studies show that none of the mutations that happen were from CRISPR itself. We have to go through mutations anyway in order to evolve as a species. And then as cancer, same kind of thing. The chemotherapy that you do have to go through is the cause of any sort of cancer. That would be your carcinogenic aspect of it, not the CRISPR itself. So those would be kind of, I guess, the downsides for some of those treatments. Other than that, really there's nothing that shows in the scientific literature that CRISPR has any direct correlation or causation to any maleffects unless deliberate.
If it has to be deliberate, is there a way that someone could like weaponize it?
I guess if you're going into like back to like designer babies, right now we are nowhere near that understanding of CRISPR yet. Like that would be a long way off. However, it could be a potential drawback, which would be like, if given to the wrong hands, could someone use it in a bad way? Yes, in theory. And that would be one of the risks. I feel like with transparency and open communication about the research, as well as maybe the legal aspects of it that would follow, would be key. I guess we would get into some problems with legal aspects, just because there could be a lot of bans, there could be a lot of restrictive legislation, and then science would kind of fall off with it. But with full transparency on the research, that we can avoid those problems.
Yeah, but full transparency implies that everyone doing this research is going to be transparent and be have a good mind about it. And I just think that is too much of a risk. I just think that's why it's unethical to use in humans.
I mean, look at all innovations over the centuries. All of it can be used in a bad way. If you take a hammer, you can use it to build a house, but you could use it to crush someone's skull. So I guess with any tool, there is a good and a bad. You just have to use it for good.
Yeah, that makes sense.
I mean, in the same vein, it's almost like a Pandora's box that we've already opened. Like, we know we can do this, and we're already doing like research looking forward that it probably can happen at some point, and we can't undiscover it. Like you were saying, be careful and open communication and all that.
Absolutely.
I think you brought up a couple good points though, and I think this could really help the human race in a lot of ways. Just almost like faster evolution. But on top of that, we're controlling it.
Yeah.
Thank you for letting me come back from the desk.
Yeah.
Really appreciate it today.
But that's all the time we have for this episode. I'd like to thank you all for watching, and if you'd like to get some Scotty Snack kombucha or see where we're getting our sources, don't forget to check the show notes or the description if you're watching this on YouTube. If you are watching this on YouTube, please do the YouTube pleasantries, like, subscribe, comment. I'd like to thank, once again, Dr. Scotty for coming on.
Thank you.
It was a pleasure to have you, but
Pleasure being here.
that's all we have for Cold Hard Truth, so we'll see you next time.